All right, thank you. I'm Jeremy Summers. I am also one of the graduate students in Nancy Chen's lab over in Rochester. I'm also there right now, just not in the lab. And I'm going to be sharing some work that I've been doing looking at the multi-generational fitness impacts of gene flow into the population at Archbold. So, the main motivation for this research here is that gene flow is crucial for small fragmented populations. Um, there are many different fitness benefits that are associated with gene flow, such as maintaining genetic variation that otherwise would be lost due to genetic drift as that population remains small. And it can also decrease levels of inbreeding in the population, which can then increase fitness. Collectively, these sort of fitness benefits are referred to as genetic rescue. And there are a few costs that get associated with gene flow as well, potentially. Um, if there's any sort of sign of local adaptation, if two populations are somewhat differentiated and they've adapted to slightly different environments, then gene flow can actually interrupt local adaptation, especially if multiple genes that have evolved together, so some alleles that are together in one population, get sort of broken up when new alleles are brought in. That could potentially reduce fitness in individuals that are outbred. And then there could also be new deleterious variation that gets brought into the population. If individuals coming from a population has more different deleterious alleles, bring them into this population, they can actually also reduce the fitness of these outbred individuals. Collectively, that's referred to as outbreeding depression. However, there's going to be differences across the different kinds of outbred individuals. So when a new immigrant enters a population and pairs with a resident, we can refer to their offspring as an F1, and there's going to be some aspects of what their genome is going to be like. Half of their genome is going to come from their resident parent, and the other half is going to come from their immigrant parent. So any alleles that tend to be different in those populations, they're going to be carrying one copy from the immigrant population and one copy from the resident population. This also means they're going to be heterozygous across many of these sites. But then if two F1 individuals pair with each other, their offspring, referred to as an F2, is still going to have about half of their genome from that resident population and a half from the immigrant population. But a major difference is that because recombination will mix and match the genomes of each one of those F1 individuals, they could be homozygous for a resident allele at one site and homozygous for an immigrant allele at another. So overall, they're not going to be as heterozygous as an F1, but they're still going to have that half-half representation. And then there's a bunch of other different crosses that could potentially happen in a population if there is sufficient immigration. So F1s could pair back to residents, and that would produce what's called a resident backcross. F1s could pair with immigrants and produce immigrant backcrosses. And if you have sufficient immigration, immigrants in your population could pair with each other and produce resident-born individuals that are entirely of immigrant ancestry, which I'm going to refer to here as immigrant offspring. So there's a bunch of different scenarios that can come from each of these different crosses that we can use to try and study how is the introduction of new genetic material through these immigrants actually impacting the population. One possibility is that this new variation functions additively. So that means that the zygote, the individual that is sort of has one copy from each population, has fitness that is in between the immigrant and the resident. So on the plot over on the right, the immigrant has potentially the highest fitness, resident has the lowest, and the F1 here is right in the middle. And that would be the case if immigrants are bringing in beneficial variation. Alternatively, there could be a major effect of dominant or recessive genes. So it's can often be the case that there's some kind of mutation that is deleterious when it is homozygous. The individual who is heterozygous won't express that deleterious mutation, and thus they're going to have the highest fitness. So we would expect the F1 individuals to perform the best. This is also referred to as heterosis and is pretty common and expected in many isolated populations. And then there's slightly more complicated effects where genes that are at different locations in the genome could interact with each other. So that's not really going to be a problem for our immigrant, resident, or F1 individuals, but for our F2 individuals, where at some points in the genome they only have resident genes, and at some points they only have immigrant genes, that allows those potential interactions to become disrupted or not behave the way that they did in their original populations. And that would lower the fitness of F2 individuals. And then finally, there could be effects that are not at all genetic, but are associated with immigrant ancestry. So if new immigrants have a social or environmental factor that impacts their fitness, such as familiarity with the environment or familiarity with your neighbors, we might expect immigrants to have different fitness than resident-born individuals. And that would include those residents of entirely immigrant ancestry. So there have been a few studies that have tried to look at similar cases in other systems. There's a really nice study of song sparrows that live on an island in the Pacific Northwest. It's a very isolated population, and they've only gotten about 40 immigrants over the entire study period of about 30 years. Um, but what they looked at is the differences in lifetime reproductive success of residents, and looking at this plot, starting at immigrants, F1s, F2s, and those resident backcross individuals. What they mainly found was that there weren't very many differences between fitness of residents in these groups, except for males, where F1 males had higher lifetime reproductive success, sort of supporting that heterosis idea, whereas F2 males had lower lifetime reproductive success, indicating there's some kind of outbreeding depression, possibly those epistatic effects. So this really showed the case that you can have heterosis, which would be, benefits of immigrant ancestry in this population, and outbreeding depression at the same time, and that it can be really specific to sex. But we need to have a lot more studies that look at this multi-generational fitness of immigrants, and especially in cases where there's much higher immigration, because increasing connectivity is frequently a conservation goal. So what actually happens if we can achieve that goal? And the Florida scrub jays at Archbold specifically make an excellent study system for this. Of course, there's many different fragmented populations across the state, but that also creates this lovely little isolated population that we can use for study. The cooperative breeding system the scrub jays allow those social effects that I considered to possibly play an important role when we're looking at immigrant fitness, because individuals interact with each other as neighbors frequently in territories, or also interact with each other within territories. And then finally, the multi-generational pedigree of the population of Archbold is crucial to be able to identify who's an immigrant and classify individuals into all those different categories. It's also especially interesting considering that Archbold has, while that population has remained stable, the immigration rate into the population has rapidly declined over the past 30 years. And that has led to a coinciding increase in the average inbreeding coefficient of breeders over time. So the first step to this project was to classify individuals into those different ancestry categories. So these are all the individuals within the demo tract at Archbold, classified into the categories, and you can even see that the bluer immigrant categories are decreasing over time. There's not that many of them in the more recent years. But it does really show that Archbold is a very admixed population. The majority of individuals have some form of immigrant ancestry. And if we even go further back to great-grandparents, almost all individuals have some form of ancestry that leads from outside of Archbold. So I took those different categories and I looked at the fitness of these individuals at different life stages using Bayesian general linearized mixed effect models. So I looked at juvenile survival, the survival of adults, and also their likelihood to establish as a breeder, and then finally their reproduction of new juveniles, finishing the life cycle. And to take it all together, I also modeled lifetime reproductive success itself. So let's go right into those results, where for juvenile survival, all the plots that I'm going to be showing are going to be presented in this sort of format where these distributions are the posterior likelihoods from the Bayesian models. Essentially, it's how likely is the estimate within this. And the bars that I'm showing is our credibility intervals for how likely is it that there's going to be a significant difference between residents and each of these different ancestry groups. So to interpret this, the female immigrant backcrosses that have that bar that's slightly to the right indicate that there is a positive trend. There is increased juvenile survival in them relative to residents, but it's not quite to the level of significance within this, or at least not at a 0.05 level. But this matches what was previously found with also the song sparrows, so it's not super surprising that we wouldn't see this effect in juveniles. When we look at annual adult survival, what's really interesting is we do find a significant increase in survival for immigrant backcross females. And what's more interesting further is that that effect is increased when we also consider the immigrant status of individuals' parents. So not included within this plot, but I also considered if your mother or father was a new immigrant rather than just an individual of immigrant ancestry, had a significant effect. Specifically, having a new immigrant mother reduced survival for these individuals. And so if you accounted for that, these immigrant backcross females had even higher survival relative to residents, considering that a good portion of them had new immigrant mothers. For transition to breeder, the groups that really stand out is the new immigrants themselves, with new immigrant females having a much higher likelihood to establish as a breeder. And this is considering new immigrant females that first establish as non-breeders and then establish as a breeder later on. Moving on to annual reproductive success, the main difference that we see here is that for F2 males, there is this decrease in annual reproductive success, sort of lining up with that outbreeding depression that was seen in the song sparrows, which is very interesting considering we don't see any other effects for males. And then finally, taking this all together with lifetime reproductive success, there's actually this positive trend still in immigrant backcross females, and a slight trend for new immigrant females as well. The plot that I'm showing here is not controlling for that parental identity, but I do have results that show that if you do control for parental identity, the effects for immigrant backcross females, immigrant offspring, and F1 individuals all become significantly greater for females. So these groups that have more immigrant ancestry also have increased lifetime reproductive success for females. So taking this all together, our results show some evidence of this sort of additive benefit for immigrant ancestry, specifically in females, but maybe these epistatic effects in males. And this behavioral effect that we might see for immigrant females when it comes to establishing as a breeder, considering we did not see that effect in any of the other groups of more immigrant ancestry. What could also really factor into this is some aspects of the territory acquisition system, with males being more likely to inherit territory from their fathers. That likely impacts the fitness of these resident and immigrant males in different ways. So overall, the main takeaways that I have in the study is that there still seems to be extremely sex-specific impacts of gene flow within this population at Archbold, which is interesting that it matches up with a distant population of a completely different species as well. And the fitness benefits of gene flow do exist despite there still being high immigration into this population. This also shows both evidence of genetic and social factors influencing immigrant fitness. So it's going to be really important to consider both the ecology of any group as well as possibly the genetics that are being brought into the population. Some really important future directions that I want to take this in is that there's variation among new immigrants. They're not all coming from the same population, and they could be more or less related to residents already. If they come from a closer population, they'd likely be more related, and we would expect different outcomes based on their relationship. So the plan is to use this genetic data that Faye was talking about to look at the heterozygosity of these immigrant descendants and see, does that really play a role? Essentially, if your parents are more closely related to residents, is that going to lessen or enhance the fitness differences that we see? And with that, I would really like to thank everyone that's made this project possible, especially everyone who works at Archbold, making this data set that is amazing and allows us to explore these really unique questions within this system. And I would be happy to take any sorts of questions on this at all. Thank you for listening to my talk.
That was a great talk, Jeremy. Thank you so much. I'm happy to relay questions to Jeremy. If anyone has questions. Fitz.
Is it possible that the result of this kind of work could help management of the forest growth days direct us to choosing which populations should serve as best served as a kind of located individual for
So Jeremy, Fitz is asking if your results in any way inform how to choose individuals for translocation and for genetic rescue of small populations.
Yes, these results do actually have some connotations for this, especially once we can look at the more genetic results and differentiate immigrants themselves. Because it'd be interesting to see if there is this difference between more closely related or more distantly related populations. Right now, these results would seem to be promising towards any sort of gene flow being beneficial to the population. But especially I'd be curious about any differences with this effect on male fitness of annual reproductive success. If that might be related to being more distantly or closely related, it might prioritize choosing closer populations for translocation or connectivity.
Rahul has a question. So first question is, this is one of two questions that Rahul has. The first is, how did you define immigrants?
Yes, so this was a very important aspect of this, which is that an immigrant in this sense is an individual who is confirmed to not be born within the monitored tracts of Archbold. So this includes the South Tract and any of the surrounding area of Red Hill and also Junkin. So within that continuous scrub habitat, all individuals born there are considered resident. Individuals that were not identified as being born there within this study period, and thankfully, since it starts at 1999, we do have the years prior to look into it, are then considered immigrants. It is very important to note that those individuals then can be coming from a really wide variety of locations.
And your second question.
The second question was, do you think some of the results you see are based on potentially females being dispersed in sex and don't?
Yeah, so the question is, how are your results affected by the fact that the females are the dispersive sex?
Yes, so I think that is a really important aspect of this, especially initially I was really concerned with female dispersal potentially interfering with the metrics that I had for survival. But even when, for example, controlling for likelihood of transitioning and also for controlling for the new immigrant status of individuals, so if individuals are already dispersed, we still see these differences. So even resident-born females of different immigrant ancestries have these differences in survival, indicating that it's not least a sort of social dispersal difference. It could potentially be if there'd be some sort of heritable aspect of dispersal distance that could play a role. And another important role in this is that because females are the more dispersive sex, there may be greater benefits to being less related to individuals that you're pairing with. So that might be why we see this greater effect in females rather than males. Males, you may get a fitness benefit to being related to where you live, whereas females, you may instead get this fitness benefit of being less related to where you establish as a breeder.
Thank you, Jeremy. There's one last question before we break for lunch.
Yes.
It's Angela Tringali asks, says, these results are really interesting. Is it possible to look for silver spoon effects based on quality of source population? And she's hinting at suburbs versus wildlands.
Oh, that would be really interesting to be able to look into it. Unfortunately, we don't have too many individuals that we have flagged as specifically coming from identifiable suburban populations. There are a few PLE known individuals, but it'd be really interesting to see if there's quality differences between there. Once we get more of the genetic data, I think that I would really like to do is even just like see if we can find any sort of structure to classify individuals. But right now, I don't know if I have the data to identify where they're coming from, or even like just bracketing the individuals of sort of how they tend to perform. But it'd be something I'd really want to look into.
Great. Well, give both Faye and Jeremy a big round of applause.